Okay, thanks, Andy, and thanks, Dennis, for inviting me. Welcome, everybody in the room. I'm going to talk about Lenia and artificial life. I'm Bert Chen from Hong Kong. Um, I'm a software and data engineer by day, and in my spare time, I do independent research in topics like artificial life or human evolution. I also make infographics or typeface. First, we will introduce artificial life or ALife and Lenia. And then we'll talk about the biology of Lenia. We'll have some further discussions and a Q&A session. We first talk a bit about the broader context about artificial life. In biology, we try to collect and study existing organisms, and we try to classify them and figure out how they work. But in artificial life or ALife, we instead simulate life or create new forms of life and try to answer questions like what is life or what life can be. We have a few approaches in what's ALife. We use synthetic biology and biochemistry to hack the DNA and create artificial cells. In hard ALife, we apply robotics and electrical or mechanical engineering to create robots and machines. Like this, you can see these humanoid robots, Atlas, that can do a backflip. Or this strand beast built by Theo Jansen. The third approach is software ALife, that we do computer simulations of life. We can talk about art in ALife. That's all these beautiful graphics installations. They are actually artworks. In fact, we can consider AI as part of ALife because things like artificial neural networks, they are inspired by the neurons in the brain, and genetic algorithms that are inspired by the process of evolution. So they are actually trying to replicate the success stories of life. We will now focus on computer simulations of life. Like other complex systems or chaotic systems, we start from very simple rules, and we can see the emergence of complex life-like patterns or behaviors. There are some classic models like Avida that simulates the evolution process, or Boyds that simulates the flocking behavior of birds. We have systems that simulate evolution to evolve new creatures, like virtual creatures pioneered by Carl Sims, or the soft robots that they can learn how to swim or how to walk. We have models like cellular automata and reaction diffusion systems that they can generate interesting lifelike patterns. And we also have particle systems that simulate chemical molecules like swarm chemistry or primordial particle systems. That's you may heard about them. Talking about soft robots, recently earlier this week, we have a big news. That's a joint team of ALife researchers that they just announced that they created the first ever living robots. They first used computer to evolve soft robots for a certain task like walking. And then they transfer the final design, the virtual design, to real frog cells. And amazingly, the cells start to walk. So it's like a crossover between soft ALife and wet ALife. So the story of Lenia begins in the 1990s, and when I got my first PC with 8 megahertz, 82, 86 CPU. And I remember this was a turbo button to turn it into 16 megahertz. So I got this PC and I used Pascal, the programming language, to write simulations of many models like gravity, strange attractors, the Mandelbrot set, and also the Conway scheme of life. The Conway scheme of life is a cellular automaton, or CA, created by John Conway. You should have heard about this before. It's basically a 2D rectangular grid of cells. Each cell has a state of 0 or 1. That means dead or alive. And each cell has a local neighborhood of its 8 neighboring cells. There's an update rule. For each cell, we count the sum of its 8 neighbors. And we use a simple if then else statement or a rule to decide the next stage of the cell. And then we apply this rule to the whole grid of all the cells, and we repeat the updating of the grid again and again, and we start to see many interesting patterns to emerge. Like this, you may see these gliders moving, and also we see we can discover glider guns that emit these gliders. We can also use these gliders and glider guns and other objects to build logic gates, and finally we can build a virtual computer inside the game of life. So it can actually do any kind of calculation in this virtual world, or we can say that the game of life is Turing complete. So at that time, I started to play with the rules of game of life. For the 0 or 1 states, how about allowing multiple values or making them floating point numbers? For the simple 8 neighbors, why not make it a wider, longer range, or make it circular in shape? It's also exploring to just count the light neighbors. So I tried to use weighted sum, and the weights are distributed as a ring or concentric rings that is equals to the calculation of convolution of the cells with a circular kernel. About the if then else update, we can use mapping functions instead, and we can make the update incremental. So all these change I made and programmed, making everything continuous, and the system no longer is no longer discrete like the original game of life. So, and then something really strange happened. I start to see this guy moving on the screen. At that time, I was still printing out numbers, so it's raw. And then I put these numbers into Excel and start to see these beautiful shapes. And then I find more other moving or rotating patterns. So what's happening here? And then we now enter the world of Lenia. I give this name because in Latin it means smooth creatures. Mathematically, it's a continuous CA in 2D or 3D or higher dimensions. The update rule can be expressed by this formula. That means we have an array of cells, A, and we apply convolution with a kernel, a circular kernel, K, and to calculate the weighted sum. And then we apply the mapping function G to calculate the delta and add a small portion of the delta back to the cells. In this model, we have a few parameters to tune. I call them mu, sigma, and beta. So far, we discovered more than 500 species of moving patterns. And they look and behave like microorganisms that we can see in some plankton videos. We start to study their structures and dynamics, and we do statistics analysis. The software is the central part of this project. I wrote several versions of the program in different languages, like the earliest version of web version. You can play it in your browser, and all the functions are there. And then I wrote a MATLAB version. It's quite powerful. And now we have a Python version that is under active development. The program acts as a kind of laboratory for doing observations or experiments on the linear patterns. It's like a portal between our world and the alien linear world. To make the program faster, we use Fourier transform, FFT, to calculate the convolution. And we also use, we can also work in GPU and maybe FPGA or TPU in the future. There's also an interactive UI that provides lots of tools for you to manipulate the cells or do numerical analysis. I'll demo the program now. So this is the Python program. This is the first life form you see in the slide. I call it Orbium. And it detects I have a GPU, so it's using GPU. I will show more about this program later in this talk. This is the data flow of the whole project, how I use the program. The main program is a linear UI that reads data from a JSON file that have all the specimens of the patterns. It generates images and allow us to observe and do experiments. I also wrote a C# program that is Lenia Auto to automatically generate lots of statistical data. And then I feed all this data into a huge Excel file and to cleanse, explore, and visualize the data. In the future, it's actually under heavy development. We will have a Lenia API that includes all these functionalities of a linear UI, linear auto, or even Excel that can be run on cloud like Google Colab Notebook. That means we can use the TPU on the Colab. So Lenia has grown into a large project. There's software that I put on GitHub, and also there's there's art. There's a showcase video that won some awards in Japan conferences. I wrote a research paper, and it's published in arXiv and Complex Systems. I'm also having talks about artificial life, and I'm starting to thinking further research in like crossover in ALife and AI. The second part is I'll have more details about the biology of Lenia with the demo program. I will show some demo to you. Imagine we discovered some alien life on Mars or on Europa or some exoplanets. For example, those ones in the novel Solaris by Stanisław Lem. This kind of symmetry. If we discover this life form, we may try to communicate with them, maybe with some musical notes, or some scientists may try to study them. These life forms should be very different from the life on Earth. They may have no eyes, maybe no arms, or they don't even use DNA, or they may be not carbon-based. But it's possibly they will have some basic principles that is common with terrestrial life, like symmetry or evolution or entropy, minimizing the entropy. So it's reasonable to use to borrow concepts or terminology from biology. We can talk about like taxonomy or ecology or the morphology of behavior, or even physiology of these life forms. The same applies to Lenia. Evolution here means how can we create new patterns or life forms in this virtual world. The term is called iterative evolutionary computation, or IEC. It's a process to manually simulate natural selection, like a mutation, selection, or reproduction. There are several ways to evolve new species. In my experience, we can randomize the grid and see if there's some random moving patterns come out from the soup. We can also tweak or tune the parameters And sometimes an existing species will turn into a new species. We can also use a program to automatically do a grid search to search for lots of new patterns. And also we can do manual mutations, like cutting a life form into half and stick it back and see if it will turn into a new species. And we can do this in this Python program. So we can randomize the grid and see if there's anything come out. Not yet. Okay, sometimes we'll see orbiums come out from almost. Or we can start from this existing life form. We can stabilize this. And we can change the parameters. Now I change sigma. And it will turn into a slightly different life form that is now oscillating. After this process of creating new patterns or life forms, we can start study the basic properties of space-time. It's like some kind of fundamental physics. The patterns, we find that the patterns are invariant with the scaling of space-time or the functions that used in the kernel or mapping, or the patterns will persist in under some transformations or deformations. So it's a very interesting phenomenon in Lenia. We can scale the space down and then scale the space up. Here, I'm not zooming the image. It's actually changing the cells array and also changing the kernel. So it's actually changing the grid and the rules, but the pattern still persists. We can also change time, like make it faster. Let's see, make it faster, make it slower. For slower time, I mean adding smaller portions of delta in the update rule. So again, I'm changing the rule. It's not fast forward or slow motion. In the kernel or in the mapping function, we can use other functions. Like now I'm using a smooth function, so the pattern is smooth. I can use step functions like this one. Oops. I can use step function, and it still persists, but the shape changed a bit. The texture changed a bit. We can also scale or rotate the pattern. And it's still here. Let's make some noise, add some noise to the pattern, and it's still here. So it's the basic properties of these patterns in Lenia. We can, it will persist even if we do such drastic changes to it. There are trillions or zillions of insects on Earth, and biologists try to group them into different taxa, like species, genus, family, or orders, etc. In Lenia, it happens that we can also apply this kind of classification to the patterns into different taxa of different ranks. If a number of samples of the life forms have, if they have continuous variations, one can be morphed into another one smoothly, then we can say we can group them into a species. Likewise, if we can group several species into, if they have minor differences, we can group them into a genus. And group different genera with similar building blocks into a family, and so on. We also give a binomial name to each species, so we can easier to identify them. Like the one I show is called Orbium unicaudatus. And so now we have a systematic classification system that we can tell which one is which. We can see in this chart on the left, there we discovered 18 families in Lenia. So they all have different structures and behaviors. We can talk about the geography or ecology of Lenia. For different life forms, they have different values of mu and sigma. That is, they are the parameters of the mapping function G. So if we try to plot these values in a parameter space, we will get a map. You can see the map here with colors and with the shape of the life forms. It shows that each species occupy a continuous area in the parameter space. And I call these areas habitats or niche. We can see that most species they occupy the central diagonal line in the middle. This line has a name. It's called the edge of chaos. That means these life forms living between the area that nothing happens and between total chaos. So these life forms actually corresponds to Wolfram's class 4 CAs. Also for the beta parameters for the kernel, we can create a beta cube that is another kind of map, and the species occupy certain areas on this cube. In the study of the morphology of Lenia life forms, we find certain prominent features, like the most striking one is symmetry. and how symmetry and behaviors are correlated. We find many kinds of symmetry in Lenia, like bilateral, radial, rotational, spherical, or spiral symmetries. With bilateral symmetry, the patterns have very fast directional movements. With radial symmetry, the life form is either slow moving or stationary or rotating. There are many life forms that are segmented or metameric. That is, they are composed of repeating components and that can make really, really long chains. Some life forms are made up of a swarm of granular masses. They are more freestyle in the structure and behavior. I'll show this in the program. So this Orbium again, we can turn the polar mode on. This is the polar mode, and it detects that it has bilateral symmetry. This one has radial symmetry. It's a pentameric, 5-fold radial symmetry. And it's stationary. This one, it has a 6-fold radial symmetry and is slowly moving. And also this one is radial, it has rotational symmetry and is rotating. I'm actually using the polar coordinates and also FFT to detect the symmetry. So it's the polar mode, and we are back to normal mode. This pattern has segmentation. It has 1, 2, 3, 4 segments. And I'll show you some more patterns. Like this one is longer, or some longer ones, like this one. This one is with the swarm architectures as granular masses is freely moving, and there's some kind of amoeba-like life form that is moving freely in the grid. One interesting thing is in nature, we also find similar relationships between morphology and their movement. In bilateral animals like insects or animals, mammals, we have fast moving. We can move fast to catch prey. In radial symmetric animals like starfish or coral or jellyfish, they are slow moving or just floating or seesaw. We see segmented animals like crustaceans and insects. Their morphology become very versatile, and They are also swarm like amoeba. So the similarity between real and virtual life forms are staggering. In the study of the behavior of life forms, we can summarize the global movements or local motion into 4 types. They are stationary, rotational, directional, and gyrating. There are also many types of local movements or gaits, like solid or oscillating or alternating. More interesting are those irregular or chaotic movements. Some of them will just wandering around without any single direction. Some of them even engage in metamorphosis. That is extremely chaotic. They switch or shape shift among different modes. There are also particle reactions. That's a very interesting phenomenon, like fusion, fission, or diffraction, etc. There are 4 types of global movement. One is this directional movement. This one is stationary, just stay here oscillating. And this is directional. This is gyrating. Also, this is rotating. So these 4 kinds of movement. About local movement, this one you see is moving, but actually is also quite solid. So it has no local movement. This one is oscillating. And this one is alternating from left to right to left. So it's like walking or will show some chaotic behavior. This one is very stable. It's a normal chromium, and this one is more chaotic. You see, it's like wandering around with different directions. And this one is shape shifting. You see now it's moving, wandering in chaotic direction. We make faster. Sometimes it will stop and change into a rotating pattern. Let's see. Yeah, now it's just changed into a stationary pattern and then change back to a moving pattern. Yeah, it now changed to a oscillating pattern. Also, particle reactions. we first load an OBM and place some more, and you will see some particle collision, and sometimes they will eat each others, and sometimes they will have other kinds of interactions. Okay, they fuse together into another life form. In biology, we have big and small variations within or between species. Like these beetles I saw in my Korean trip, there are very subtle variations inside a species. The same is true in Lenia. So we want to do quantitative analysis on the structures and behaviors to see the subtle variations. First, we decide which kind of measurements to take, like mass, size, shape, linear speed, or angular speed, etc. And then we calculate and collect these measurements for lots of life forms. With lots of this statistical data, we can now plot graphs like this one on the top is mass versus the parameter sigma. Now we can discover some trends or subtle variations among these life forms that they actually change into another species when we change the parameter. Also, this chart at the bottom, I plot speed versus mass, and we can discover some interesting correlations between these measurements. It's also possible to do advanced statistics, like just now we showed that you can detect symmetry and also detect the oscillating period and also chaoticity. This one is gyrating orbium. So this is the statistical mode. You can see this mass and growth change over time that create a phase-space trajectory. We can change to other statistics like size. This is some distance. And this is speed. And this is angular speed. There is periodogram that is used to detect the period of this pattern. So it detected its period is 0.87 second in this virtual world. Now we will have some further discussions about Lenia and artificial life. With the discovery of Lenia and also smooth life, we can now classify this cellular automata by the level of continuity. We have the original CAs, the discrete CAs, like the game of life. There are geometrical CAs like Lenia. There are also continuous CAs like real life. In discrete CAs, the patterns are very precise, but also very fragile. That means they are easy to be disrupted. We change one cell and the pattern is gone. The patterns are also digital. That means some rules can literally do calculations thanks to the Turing completeness. In geometric CAs, patterns are fuzzy but resilient. They can persist from changes or collisions. They are also analog and lifelike. The real continuous CAs are purely mathematical models. If we scale up Lenia so that the kernel goes to infinitely large, then we will approach the continuum limits of a truly continuous CA. So continuous CAs is actually, we can't do simulation, but we can approximate it by things like Lenia. We find quite a few similarities between the Earth and terrestrial and Lenia life. They are both vivid, eye-catching, and actually quite beautiful. Like this video, we show some calm jellies, a video from aquarium, and also a life form in Lenia. I called calm bug. The both exhibit great biodiversity. The patterns or the life forms are exhibit phenotypic plasticity. That means they all can adapt to changes and undergo evolution. I have a hypothesis that it seems like symmetry provides stability, and asymmetry in the structure provides motility, the ability to move. So I think these rules can be applied to both the real life forms and the virtual life forms. The ultimate question is, what is life? How can we define life? One may say that, okay, I know it when I see it. I think this statement is actually true because so far the human brain is the best life detector. We can know this is life when we see this structure and we see it moving. So I think we can trust the human brain in this task, but more precisely, we can define life by a couple of capabilities like self-organization, self-regulation, self-replication, etc. Lenia exhibits a number of these capabilities. So can we say that the patterns in Lenia is alive, or are they partially alive? The same situation happens in astrobiology or virology. Can we say that a virus is alive? Or when we see some objects in space that is moving in some peculiar way, and how can we determine that these space objects are alive? These are all subject to debate. Here are some future directions to ask or to we can work on. I want to answer a few questions that is related to cellular automata. Like, is there any self-replicator in Lenia, or is there any emitter of patterns like those glider gun in the Game of Life? And is Lenia Turing complete? Can it used to perform computation? We can explore some variations like higher dimensions or parallel universe. I'm now making a API and probably a data set. They can be used to do data science or machine learning using Lenia as a substrate to do more research. And based on that, we can start to think how to do crossover research between ALife and AI. For example, how can we apply AI methodologies like genetic algorithms or deep learning to do object detection or automated search on a ALife system. We start to see some 3D and 4D patterns. That's very interesting, but let's see, can be shown. But you can see them in my Twitter. I want to introduce some related works in the literature or some is now in progress. Stephen Raffler's Smooth Life. You see the picture in the top. This is also a continuous cellular automaton discovered a few years ago. Is very similar to Lenia, and it's actually a case of independent discovery that we arrive to the similar system using similar principles, starting from the Game of Life. On how to do automated search of patterns, we have P.Y. Udayar's curiosity-driven goal exploration algorithm. We have Ken Stanley's neural evolution and novelty search, or the algorithm called NEAT, that should be very useful. And David Haas works on neural evolution in deep learning. Alex Motvinstev work on NeuroCA is in progress. He make a hybrid of cellular automata and neural networks that you can see the bottom image. It can it simulates some kind of regeneration in the grid. And Nick Kuparisa's work, he is doing a FPGA chip for calculating cellular automata faster. If you want to know more about artificial life, there are a few conferences or communities. In mid-July, there will be an ALife conference in Montreal. The main theme is what can ALife offer AI? So it will be very interesting. And there is a small community in Lausanne called Quine Association. It explore topics like artificial life or quines or creativity. That I meet its members a week ago in Hong Kong. I also made a Twitter list of artificial life news. So we can go to these URLs to see these things. That's all, and thank you for listening. And by the way, 香港人加油。Yeah, that's the Q&A. Any questions?
I'm getting this set up and pairing the microphone. Come on. Can you hear me now?
Yeah, I can hear you.
I'm the Verizon guy now. Okay. Anybody questions? Okay. Yeah. Actually, I saw the slash dot thing on the frog, and I was going, "Whoa, that's a little too creepy for me today."
You mean these patterns?
Yeah. The frog cells, and they were convincing.
Oh,
to do stuff.
okay. Yeah, these frog cells.
Yeah,
they call it a programmable organism. Is actually, as I know, is in the very first stage. So it's kind of you have to do make the cells by hand. Mm-hmm. It's not ready for mass production. So it's not that kind of people will start to thinking about Terminator or Frankenstein, but actually it's not already there. They are making progress, but the progress is very interesting, I guess.
Yeah, I know it's. Oh.
Have you used anything like evolutionary algorithm or other more complicated search algorithms other than grid search to try to generate these life forms? Because I saw that you had done a few different kinds of like randomness and grid search, but I was thinking that maybe some kind of like more specific kind of search algorithms may give you different or new life forms.
Did you hear the question?
Yeah, I heard it.
Okay.
Yes. The method I use is called interactive evolutionary computation. That's a methodology inside evolutionary computation. So it's actually using some kind of manual process to simulate the natural selection. The process is not automated, so it needs feedback or needs manipulations from a human user. So I'm planning to study other kinds of automated search program mechanisms and apply to Lenia, like those ones I mentioned. The works in the genetic algorithms or evolutionary algorithms. They have so many methodologies out there that I have to try to apply to this system. So let's see what happens.
Cool. I think we're good. Well, thank you very much. And
Okay.
yes, it's daylight there. Last speaker was not daylight where they were. So cool. Thank you.
Okay, thank you.